All right, welcome back. For today's deep dive, we have a fascinating set of materials to unpack. It's all about, well, how we know if our tests are actually giving us reliable results.
Exactly. It's all about trust, isn't it? And particularly this time, we're zeroing in on analytical method validation in the pharmaceutical industry.
Yeah, the stuff that underpins basically every decision in drug development, right?
Absolutely. We've got excerpts from books on drug development, some key regulatory guidelines, and other materials you sent over.
Yeah, a really interesting mix.
Yeah, and I think our mission here is to sort of demystify this whole topic. I mean, method validation can get pretty technical, right?
Oh, definitely.
But it's so crucial, and it has such a huge impact in the real world. So we want to break it down, make it accessible, without getting bogged down in jargon.
Exactly. So let's start with the basics. Where does this validation process even begin, and why is it so important? Like, why do we care so much about making sure our tests are reliable?
Well, I'd say it starts right at the beginning, when you're designing the analytical method itself.
Okay, at the design phase.
Right. But the why is really about, as I said, trust. Every decision we make in drug development and quality control, it all relies on data from analytical tests.
Right, right.
So validation is like the foundation. It's what gives us that confidence that the results we're seeing are actually accurate and consistent. Without it, well, we're basically making decisions in the dark.
Yeah, that's a scary thought. So it's about making sure the tests, the tools we're using to analyze drugs are actually measuring what they're supposed to measure and doing it consistently.
Precisely, because if a method isn't validated, we just can't be sure if the results are real. I mean, think about the consequences. You could have incorrect dosing information in a clinical trial.
Oh, wow.
Or, even worse, you could end up releasing a drug that's substandard or even dangerous.
Right. Yeah, that's a huge deal.
It is. And for you, as someone who wants to be well-informed, understanding validation is so important. It's about knowing that the data we're using to make these crucial decisions is solid, it's dependable.
It's like cutting through all the noise, making sure the information is actually good.
Exactly. So once we have a method designed, what are the things we actually look at to validate it? I know there are a bunch of parameters mentioned in the materials.
Yeah, there are. So let's break those down one by one. What about specificity? What does that mean in this context?
Okay, so specificity is all about making sure the method is really only measuring what we want it to measure. The analyte, which is usually the drug itself.
Right.
And we need to make sure it's not picking up signals from other stuff that might be present in the sample.
Okay, so like what kind of stuff are we talking about?
Well, it could be impurities that form during the manufacturing process or even during storage. Or it could be degradation products of the drug itself, or even the inactive ingredients, the stuff that makes up the tablet or the capsule. We call that the matrix.
The matrix, right. Got it.
So specificity is about making sure we're not fooled by any of those other things. We want to be absolutely sure that the signal we're seeing is coming from the drug and nothing else.
Right. So it's like if we're testing a tablet to see how much of the active ingredient is in it, we want to make sure our method is only measuring that ingredient and not getting confused by the fillers or binders or whatever else is in the pill.
You got it. It's about having a truly selective method.
Okay, that makes sense. And then we have accuracy. I feel like that one's a bit more intuitive. It's about making sure we're getting the right answer, right?
Yeah, you're on the right track. Accuracy is about how close our test results are to the true value, the actual amount of analyte in the sample.
Okay.
And we often demonstrate this by using reference standards, samples where we know exactly how much of the drug is present.
Ah, okay.
And we see how well our method can measure it. And typically, accuracy is expressed as a percentage recovery.
So if we expect to find 10 milligrams, and our method consistently measures, say, between 9.8 and 10.2 milligrams.
That would be a good indication of good accuracy.
Okay, so accuracy is like hitting the bullseye, and then there's precision, which I always get these two confused, but they're not the same thing, are they?
No, they're definitely distinct, though closely related. Precision is about how close multiple measurements are to each other.
Okay.
So it's not about whether the average is exactly right, that's accuracy. Precision is about the consistency of the method. And there are a few different levels of precision we look at.
Okay, yeah, break those down for me.
Sure. So the first one is repeatability. And this is the precision we see when the same analyst, using the same equipment, runs the test on the same sample multiple times, all in a short period of time.
So like back to back to back, same everything.
Exactly. It's how consistent the results are under these really tightly controlled, ideal conditions. It's sort of the baseline level of precision.
Okay, got it. And then we take it up a notch, right?
Right. So the next level is intermediate precision. And this is where we start to introduce some real-world variability.
Okay, how so?
Well, we might have different analysts running the test, or we might use different pieces of equipment within the same lab, or even run the test on different days.
Right, things that would normally happen in a lab setting.
Exactly. And this helps us understand how robust the method is to these normal variations that occur within a single lab.
So it's still within the same lab, but we're just making it a bit more realistic.
Exactly. Then we have reproducibility, which, as you said, takes it to the next level by involving different labs altogether.
Right, so now we're talking about, like, if a method is going to be transferred from a research lab to a manufacturing facility,
Yeah.
or maybe even between different companies.
Exactly. Reproducibility is about seeing how consistent the results are when the method is used in different places. And interestingly, the ICH Q2R1 guideline, which is kind of the Bible for analytical method validation,
Yeah, ICH, that comes up all the time in these materials.
it does, and for good reason. Anyway, it mentions that if you've really nailed down reproducibility, you might not even need to worry about intermediate precision.
Oh, interesting.
Yeah. But in practice, particularly when you're transferring a method, the receiving lab will usually do their own intermediate precision studies.
Okay.
And both labs will generate reproducibility data just to be absolutely sure everything is consistent across locations.
That makes a lot of sense. So we've covered specificity, accuracy, precision. What about sensitivity? Our sources mention things like detection limit and quantitation limit. Can you explain those?
Yeah, those are key parameters, especially when we're dealing with really small amounts of the analyte. So the detection limit, or LOD, is the lowest concentration of analyte that we can reliably detect. We can say it's there, but we might not be able to accurately measure how much of it there is.
Okay, so it's like we know we're picking up a signal,
Yeah.
but it's maybe too faint to get a precise reading.
That's a good analogy. It's about the sensitivity of the method. And then the quantitation limit, or LOQ, is a step up from that. It's the lowest concentration we can not only detect, but also quantify with acceptable accuracy and precision.
So it's like LOD is about knowing something is there, and LOQ is about being able to say, okay, there's this much of it.
Precisely. So LOQ is really important for things like measuring the amount of active ingredient in a drug product, while LOD might be more relevant for detecting impurities, which we usually want to keep at very low levels.
Got it. So those are all about sensitivity. What about linearity and range? What are those referred to?
Well, linearity is about how the response of the method changes as the concentration of the analyte changes.
Okay.
So ideally, we want the response to be directly proportional to the concentration. Think of it like a straight line on a graph.
Oh, okay.
The signal goes up in a nice linear fashion as the concentration increases, and the range is simply the interval of concentrations over which we've demonstrated that the method is linear, accurate, and precise.
So we're not just checking linearity at a single point, but over a range of concentrations that we might expect to encounter.
Exactly. We need to know the method is reliable, whether we're looking at a high dose of the drug or a very low concentration of an impurity.
That makes sense.
Mm-hmm.
And then there's robustness. What does that mean in the context of analytical methods?
Robustness is all about how well the method can handle small changes in the conditions. Like, what happens if the pH of a solution is slightly off, or the temperature isn't exactly what it should be?
Right. Things happen in the real world.
They do. So a robust method will still give you accurate and precise results, even with these minor variations.
So it's kind of like how resilient the method is to the imperfections of everyday lab work.
Exactly. It's a measure of its reliability in the face of real-world variability. And finally, we have system suitability, which is really all about making sure our equipment is up to the task.
System suitability. Yeah, I was wondering about that one. It sounds like it's more about the instrument than the method itself.
In a way, yes. It's about making sure the entire analytical system, the instrument, the columns, the reagents, everything is working properly before we run our actual samples.
Okay, so it's kind of like a pre-flight check.
That's a good analogy. We use standard samples to verify that everything is functioning correctly. We look at things like the resolution between peaks, the reproducibility of injections, stuff like that.
So it's like before we set out on our analytical journey, we make sure the car is running smoothly.
Precisely. And all of these parameters we've talked about, they're not just scientific concepts, they're also heavily regulated.
Right, right. ICH again.
Exactly. The International Council for Harmonization, or ICH, plays a huge role in setting standards for the pharmaceutical industry.
And they have specific guidelines for analytical method validation, right?
They do. ICH Q2R1 is the key one. It provides detailed recommendations on how to validate analytical methods for different applications, like identity testing, assays, impurity testing, and more.
And following these guidelines is important for regulatory submissions, right?
Absolutely. When a company wants to get a new drug approved, they have to submit a ton of data to regulatory agencies like the FDA.
The FDA, right?
And that data has to be generated using validated methods. So, following ICH guidelines helps ensure that the data will be accepted by regulators worldwide.
It's like speaking the same language,
Yeah.
making sure everyone's on the same page.
Exactly. It streamlines the whole process. And of course, regulatory agencies like the FDA, they have their own specific expectations as well. I mean, we don't need to get into the nitty-gritty of regulations like the Code of Federal Regulations right now.
Yeah.
But the point is, method validation isn't just about good science, it's also a regulatory requirement.
Okay. So we've talked about the what and the why of method validation and the regulatory aspects. But what about the real-world impact? How does all this translate into actual outcomes for patients?
Well, first and foremost, strong method validation is absolutely essential for getting new drugs approved. When a company submits an application for a new drug, they need to demonstrate that the drug is safe, effective, and of high quality. And that all relies on analytical data. If the methods used to generate that data aren't properly validated, well, the regulators are going to have a lot of questions. It can delay the approval process or even lead to rejection.
So good validation is basically a prerequisite for getting a drug to market.
It is. Regulators need to be confident in the data, and validation provides that confidence. And beyond approvals, robust validation can also help prevent compliance issues down the line. If a company's methods are found to be inadequate during an inspection,
Uh-oh.
it can lead to warning letters, investigations, delays, all sorts of headaches.
And imagine that if the methods aren't reliable, it could even lead to, like, product recalls.
Absolutely. If a bad batch of drug gets released because of faulty analytical testing, the consequences can be serious. You could have patients taking a drug that's not what it's supposed to be, and that's a huge risk.
Right. That's where the real-world impact really hits home.
It does. And while the specific sources we're looking at today don't have a lot of detailed examples of, like, specific cases of validation successes or failures, it's important to remember that everything we've talked about today, all these parameters, all this rigor, it all feeds into the bigger picture of drug development.
Yeah, it's the foundation.
Exactly. And when you see a new drug get approved or a company consistently producing high-quality medicines, it's because of things like analytical method validation.
It's kind of like the unsung hero of the whole process.
It really is.
Well,
So to sum it all up for our listeners, analytical method validation is a fundamental process in the pharmaceutical industry. It's about proving beyond a doubt that the methods we use to test drugs are reliable and accurate. It involves a whole bunch of parameters, it's heavily regulated, and it has a huge impact on the quality of the medicines we all rely on.
yeah, this deep dive has definitely been eye-opening. I feel like I have a much better understanding of why this seemingly technical area is so important.
I'm glad to hear that. It really is crucial. And, when you think about the level of scrutiny and rigor involved in validating these analytical methods, it really speaks to the level of care that goes into developing new medicines.
It does. It's a reminder that there's a lot happening behind the scenes to ensure that the drugs we take are safe and effective.
Exactly. And we hope this deep dive has shed some light on that process for you. If you have any questions or if there are other areas you'd like us to explore in future deep dives, please don't hesitate to let us know.
Yeah, we're always happy to go deeper. Thanks for joining us for this one.
It was my pleasure. Thanks for having me.
All right, until next time.
See you then.